Hi everyone, we're S-Cube, the science student organization from university. I'm Rachel, the president.
Hi, I'm Nicole, the internal secretary.
Hi, I'm Sarah and I am the Secretary General.
And I'm Julia, the vice president.
And today we'll be performing an experiment to extract DNA from a strawberry.
Now, as we all know, all living beings have DNA. It is the blueprint for all that happens inside a DNA cell.
So during today's DNA extraction, we will be using a detergent, which will cause the cell to pop open so that the DNA is released into the solution. Then we will add alcohol to the solution, which will cause the DNA to precipitate out.
In this activity, this is exactly what we'll be doing. We'll be using strawberry because of their 8 copies of genome in each cell. This is a whole lot of DNA for each cell, which will allow us to see the DNA more clearly when we actually carry out the experiment.
So for this experiment, you will need some strawberries, a Ziploc bag, a sieve, tweezers, a beaker, an empty beaker, some water, some dish soap, some chilled ethanol, and some salt.
So first, we leave the ethanol to chill, and then we have the 90 milliliters of water in a beaker, and then we have to add the 10 milliliters of dish soap into the water. Then I am going to add the 1/4 of teaspoon of salt into the beaker, and then I am going to mix it all.
So what you need to do next is open the Ziploc bag and place just 1 strawberry inside. In addition to this, we will add the solution that we have previously prepared. Make sure that the Ziploc bag is sealed tightly and make sure to let as much air out as possible. Then what you need to do is to mash the strawberry until no chunks or large pieces are seen. So once you're done with this step and you can see that there are no large pieces of strawberries left, you must open the Ziploc bag and take an empty beaker and a sieve and pour the solution inside the beaker. Then, using a spoon to help you, stir the solution so that the liquid separates from the solid parts.
So by adding the salt and the detergent mixture to the smashed strawberries, the detergent helped to pop open the strawberry cells, releasing the DNA into the solution.
Now, the salt, on the other hand, is helping to create an environment where all the different DNA strands can clump together, allowing us to see them better. Back to the experiment.
So now we are going to add 5 milliliters of iced ethanol.
Now, what the cold rubbing alcohol does is precipitate the DNA out of the strawberry liquid, while the rest of the liquid stays in the solution. What should now start to form are white gooey DNA strands in the alcohol layer and just beneath it.
So a single strand of DNA is very small, so small that we cannot see it with the naked eye. However, due to the number of genome copies per cell in a strawberry, remember there are 8, and because they are clumped together, we will be able to actually see it.
Now, as you can see, all those tiny little dots are actually DNA strands. If I can just pick one up, I'm not sure if you can see it, but these are the DNA strands from just one single strawberry.
So that was the experiment. I hope you all learned something new today, and I all hope that you enjoy the rest of Science in the City, and see you next year.